Hi, this is Matt Dean with A+ College Ready. Today we're going to talk a little bit about embryonic development. And we're going to talk about it typically in terms of human embryonic development. So embryonic development begins when a haploid egg, which we might notate as N, that means it has one copy of each kind of chromosome, and a haploid sperm fuse together to form the organism's first diploid cell, the zygote. Remember, diploid we're going to notate as 2N. Meaning that cell now has two copies of each type of chromosome. The cell then goes through a period of between 12 to 24-hour period of rapid cell division. In this case, it's doing mitosis over and over again. This rapid period of cell division is called cleavage. During this stage, cell division occurs so rapidly that the cells really don't have any time to grow. They're just multiplying. When the developing organism reaches the 32 cell stage, it's called a morula. This ball of cells is about the same size as the original zygote. It now has, though, 32 cells instead of one. The morula consists of a solid ball of cells. It's still surrounded by the zona pellucida, which was sort of the protective coating around the original egg cell. At this point in development, all of the cells are called totipotent cells. They're totipotent stem cells. This means that these cells still have the potential to become any type of cell in the embryo or in the extraembryonic tissues, the cells that will become the placenta. This is because that essentially none of the genes yet have been deactivated by methylation or other factors. By about day 4 of embryonic development, the cells continue to divide, but they also begin to differentiate. During this stage, which is called blastulation, 2 layers of cells develop. An outer shell-like layer called the trophoblast. This layer eventually becomes the placenta, and an inner mass of cells, ultimately called the inner cell mass. These cells will ultimately form the embryo. The inner cell mass gets pushed off to one side of the structure, while the rest of the inside of the sphere forms a fluid-filled cavity called the blastocell. At this point in development, the entire ball of cells is called the blastocyst in mammals, or the blastula in non-mammal animals. During this blastula stage, the cells of the inner cell mass are called pluripotent stem cells. This means that the cells have begun the process of differentiation and that some of the cells have been deactivated, probably through methylation. Pluripotent cells are still capable of becoming any type of cell in the embryo, but at this point they now lack the ability to become the extraembryonic cells of the placenta. So here we see the egg and the sperm getting together through fertilization. That makes the zygote. Those cells divide multiple times during the process of cleavage to form the morula, the 32 cell stage. Those cells are totipotent. They can still become any type of cell either in the embryo or in the extraembryonic tissues. Eventually those cells organize into layers, the trophoblast that you see on the outside and the inner cell mass on the inside, and the cells begin to differentiate. They begin to be methylated so that those inner cell mass cells are now pluripotent. They can form any kind of cell in the body of the embryo, but they can no longer form the cells of the placenta. Ultimately, during the process of embryonic development, more and more methylation happens, and most of the cells, by the time the embryo is ready to be born, are unipotent. And this means that the cells can now only become one specific type of cell. So at that point, they're not stem cells anymore. So during about week 3 of embryonic development, the cells in that inner cell mass go through the process of what's called gastrulation. During gastrulation, the inner cell mass cells organize themselves into 3 distinct layers known as the germ layers. The cells in the outermost germ layer, called the ectoderm, they eventually go on to form the epidermis, the outer layer of the skin, the hair, the nails, the brain, the spinal cord, and the peripheral nervous system, the nerves that branch off of the brain and the spinal cord. The cells of the middle germ layer, the mesoderm, they go on to form the muscles, the bones, the connective tissues, the notochord, which will ultimately help to make the spinal cord, the kidneys, the gonads, and the circulatory system. And then the cells of the innermost germ layer, known as the endoderm, go on to form the epithelial lining of the digestive tract, the GI tract, the stomach, the colon, the liver, the pancreas, the bladder, and the lungs. Once the cells of the inner cell mass are organized into 3 distinct germ layers, the entire ball of cells is called the gastrula. Also during gastrulation and invagination, an opening forms into the gastrula, and that opening is called the blastopore. In protostomes, which are animals like worms, mollusks, and arthropods, which includes insects, this opening, this blastopore, eventually becomes the mouth. Protostome, the word means mouth first. In deuterostomes, animals like echinoderms, starfish and sea cucumbers, and chordates, which are vertebrate animals essentially, this opening becomes the anus. The word deuterostome actually means mouth second. So after gastrulation, the cells undergo a process called neurulation. During this process, the notochord forms and causes the ectoderm to bend itself into a tube known as the neural tube. This neural tube will eventually give rise to the brain and the spinal cord. The cells of the mesoderm go ahead and give rise to the kidneys, the gonads, the adrenal glands, the blood vessels, and the muscles of the organs. And at the same time, the cells of the endoderm roll themselves into a tube known as the digestive tract. The organs of the gastrointestinal tract, the GI tract, start off as outpouchings of this digestive tract. The endoderm will also ultimately give rise to the lungs. So here we can kind of see a summary of the process. So we started off as a zygote that goes through rapid cleavage until eventually we get to the 32 cell stage, solid ball of cells called the morula. The morula hollows itself out into a hollow ball of cells called the blastula. The cells of the inner cell mass organize themselves into 3 distinct layers, and a blastopore starts to form. This is the gastrula stage. And here we can see that blastopore pushing further and further into the gastrula. Here's another look at it. So here's our morula, of it ultimately leading to the blastocyst or blastula. By the time we get to the gastrula stage, we have 3 germ layers inside. You can see those labeled: ectoderm, mesoderm, and endoderm. And we don't see it yet here, but we're also going to form the blastopore, which will ultimately lead to either the development of the mouth or the anus. One other thing we want to talk about briefly are the Hox genes. Hox genes are a group of related genes that help to control the basic body plan of an embryo along its head-to-tail axis. After the embryonic segments have formed, the Hox proteins determine the type of appendages, legs, antennae, wings, and so on, or the different types of vertebrae in humans that will form on a segment. Hox proteins confer segmental identity, but they don't actually make the segments themselves. A good analogy for the Hox genes can be made to the role of a play director that calls which scenes the actors are going to do next. If the play director calls the scenes in the wrong order, the overall play will be presented in the wrong order. Similarly, mutations in the Hox genes can result in body parts and limbs that end up in the wrong place along the body. Like a play director, the Hox genes don't act in the play or participate in the actual limb formation. They just help to coordinate the process. The protein product of each Hox gene is a transcription factor for the genes that ultimately make the body parts. Each Hox gene contains a well-conserved DNA sequence known as a homeobox. In many animals, the organization of the Hox genes on the chromosome is the same as the order of their expression along the anterior to posterior axis of the animal's body. And we can see that here. So here are where the Hox genes are expressed, and you can see the color coding, and here is the chromosome along which they lie. And notice the expression in the organism pretty much mirrors the order of those genes in the chromosome or on the chromosome. Now that's in a fruit fly. Here we can see similar patterns in a baby human. Here we have the genes along the chromosomes color-coded to the order in which they're expressed in our vertebrae. So again, Hox genes are very important in determining the basic body structure of an organism. They are well conserved across many species, very important in determining the shape of our bodies. All right, that's it for our embryonic development screencast. Hope that helps.